Hello, Paul Taft here, and this edition to How to Survive Your National Exams, we are going to be talking about blood tubes. Blood tubes play a critical role in phlebotomy, and knowledge of them is sort of fundamental in your ability to become a rock star phlebotomist. So how do you do this? It takes a little bit of practice, but I'm here to show you 8 simple tricks to help you master your blood tubes techniques, starting with the order of draw all the way up through tubes additives. When you are working in an inpatient or an outpatient setting, it really doesn't matter where you work, things are always going to be a little bit different. This is specifically designed to help you get through your national exams because they sort of ask some very specific things. Now, where you work, one tube might do something that another tube you thought would do something doesn't. So it's just kind of backwards and forth with that. But how do you know what to draw when you need to draw it? It's mapped out for you. Your requisition will clearly state and show you the color of the tubes and the order they need to be drawn in. Pretty easy, right? Well, what if you are in an ER situation and the doctor says that he needs a coagulation, CKMB, myoglobin, troponin, stat? You might not have enough time to draw up an order and pull up the tubes and everything like that. You might just have to grab some tubes, think quickly on your feet, and draw some blood. So how do you do this? Very simple steps. Let's start with step number 1. This is the order of draw. You have yellow, light blue, red, green, lavender, and then gray is your last tube. So you can think of a little mnemonic in your head. I have one for you. Yucky little boys rarely get lovely girls. This is pretty easy to think of because it's generally probably pretty true. So now we can associate the colors with this little mnemonic with the colors of the tubes. So we have our yucky yellow. Little boys always like light blue, rarely red, get some green, lovely lavender, and girls gray. So now we have yucky little boys rarely get lovely girls into yellow, light blue, red, green, lavender, and gray. We also need to think about the departments these tubes belong into. So our yucky yellows go to microbiology, our little boys light blue goes into our coagulation, and our reds and greens are in chemistry, our lovely lavender goes to hematology, and our gray is also a chemistry tube, but it's just a little bit different in how the gray tubes work. We will get there next. Step 2 is when you're studying for these tests that go into the tube for your national exams, forget about the chemistries because there is way too many of them. Here's the simple trick. What you're going to do is memorize the tests that go into the microbiology tube, the yellow tube. Memorize the coagulation, the light blue tubes. Skip the chemistries entirely over here. Go straight to your lavender hematology, and then end with your gray tube. So totally skip the chemistries. What goes into microbiology? We're collecting blood cultures to test for septicemia. So memorize those things. Now come over to your coagulation tube, partial thromboplastin, activated partial thromboplastin, prothrombin, D-dimer, and fibrinogen degradation products. So you've memorized those 2 tubes. Skip chemistry, go on to hematology. We have our complete blood count, which includes red blood cells, white blood cells, thrombocytes or platelets, and reticulocytes. That's a complete blood cell count. We also have our erythrocyte sedimentation rate, otherwise known as our ESR or SED rate. We have our hematocrit hemoglobins and sickle cell. So we've got the yellow, light blue, and lavender down. We got to move to the gray. Gray is pretty easy. Think of gray goes with glucose. Triglycerides is one of the things that comes up, and GLYC, GLYC, means glucose. So you can test for triglycerides and glucose in the gray tube. But there's also this other one, alcohol. You can test blood levels of alcohol with the gray tube. Now you have these 4 tubes memorized. Why are we only memorizing these 4 tubes? Because literally every single other test question that you are going to get goes into a chemistry tube. There are hundreds of different chemistry tests. We don't know where they're going to go, what they're going to go into, and sometimes we won't even recognize them. In class, you might not even talk about them. Who knows what digoxin is? Have you ever heard of rheumatoid profile? Maybe, but you know that it doesn't belong into a yellow, light blue, lavender, or gray tube. It belongs to a chemistry tube. So our step 3 here is to differentiate between the chemistry tubes. We have 2 types of chemistry tubes here. We have a serum and a plasma sample. We'll start with our serums. We have our non-SST. SST stands for serum separating tube. So these are the tubes that do not separate serum. They do allow the blood to clot. We have our glass red non-additive tube, which will allow the blood to clot in about 30 to 60 minutes. And we have our plastic clot activating tube, which is a little bit quicker. Our blood will clot in about 30 minutes or so. And then we have our serum separator tubes, or our SSTs. We have our tiger top, otherwise known as the gray-red speckled top, and our gold tube. Now you can see down here, this includes a thixotropic separator gel. This is what separates the blood cells from the serum portion of the blood. So that's how you know it's a separating tube. This is an SST, serum separating tube. Now we have a subcategory of separating tubes in the serum category called a rapid separating tube. Now all these other tubes take anywhere between 30 to 60 minutes to clot. That means you can't put them in the centrifuge for 30 to 60 minutes. So with a rapid separating tube, this tube has the additive thrombin in it, which will allow the blood to clot in about 5 minutes, allowing us to get a stat serum chemistry sample. Now, you may never see this tube in the real world at all, but this tends to come up on tests, so commit this one to memory. Now we're going to move over to our plasma side, where we have PSTs and non-PSTs. PST stands for plasma separating tube. You can see down here at the bottom we have our thixotropic separating gel. You can tell that this is a separating tube because of the gel at the bottom of this. Now this light green is lithium heparin, lithium for light. Now sometimes it could be a little bit different looking. You might not have the thixotropic separating gel in the lithium heparin. It doesn't always look like this. However, you want to pay attention to these lines, these oblique lines that you see over here in the other tube, because this will indicate that it is a heparin. If it doesn't have a separating gel in it, you will see these oblique lines. So it could be lithium, could be sodium. Just make sure that you're reading these tube labels. Okay, moving on to step 4. Now this is where it gets a little bit tricky. We've memorized our microbiology, coagulation, chemistry, hematology, and our grade tubes. So we know the yucky little boys rarely get lovely girls order of draw. The next most important step is to try to think about the order of draw in terms of the additives inside the tubes. This is a little bit tricky. Now, we've been showing you this picture of this yellow tube over here. In all reality, you might not ever see a sodium polyethyl sulfonate yellow tube like this. It is sort of rare, so just keep that in mind. What you'll typically end up seeing are these larger culture bottles. The one in the center here is for pediatrics, but these culture bottles are generally in aerobic or anaerobic solution. And this allows the bacteria inside the blood, if there is any, to grow. It's like this nutrient broth, and it allows the bacteria to grow and thrive and multiply exponentially. So this is our blood cultures. Now, the blood cultures, whether or not it is the glass yellow tube or these larger plastic culture bottles, it has an additive called sodium polyethyl sulfonate, otherwise known as SPS. And this is our nutrient broth additive for the microbiology. Over in our coagulation, we have sodium citrate for our light blues. And then we have our clot activators that may or may not include the separating gel. This is our serum sample. Over to the next category in chemistry, we have our lithium heparins and sodium heparins. So lithium light green, sodium dark green heparin. And then we move over to hematology with our EDTA. This is short for ethylene diamine tetracetic acid, 2 words. And then we move over to our gray tube, which includes 2 additives. The anticoagulant portion of this tube is the potassium oxalate, but we do have another additive, sodium fluoride. So this tube is very special in that the potassium oxalate prevents the blood from clotting, so we have an anticoagulant, and the sodium fluoride prevents the breakdown of glucose. Now all these tubes can collect some measure of glucose, whether it be large or small, but it doesn't last a whole lot of time because glucose tends to break down inside the blood. So the sodium fluoride prevents the breakdown of glucose, otherwise known as glycolysis. So we can test glucose in this tube for 24 hours at room temperature or up to 48 hours fridge to frozen. So let's rehash real quick. We learned the order of draw in the colors. Yucky little boys rarely get lovely girls, yellow, light blue, red, green, lavender, gray. We know our microbiology, coagulation, chemistries, hematology, and then chemistries. So we've memorized that portion of it. Now our step 2 was to forget about the chemistry tests when we are trying to memorize the tests that go into the tubes. So we know that the microbiology can test for blood cultures and septicemia. Our coagulation light blue is PT, APTT, D-dimer, and FDP. Our hematology, our lavender tube, can test for CBCs, ESRs, hematocrit hemoglobins, and sickle cell. And our gray is for glucose and blood alcohol. Because everything else goes straight to your chemistry tubes, now all you have to do is step 3, differentiate between your chemistry tubes. Which is serum and which is plasma? And every other question you get, Which of the following tubes would you draw a hepatic panel in? Okay, well, hepatic panel wasn't in our list of tests in our yellow, light blue, lavender, or gray, so we know that it's going to go somewhere in our chemistries. So all you have to do is differentiate now. Are we drawing a serum hepatic panel or a plasma hepatic panel? Now, we need to know the order of draw by the additives. This is probably the most difficult part, but fundamentally the most important step. So our additives in microbiology are sodium polymethyl sulfonate. For coagulation, we have sodium citrate. In our serum chemistries are clot activators, serum separator tubes, rapid separator tubes. And remember, this orange guy has thrombin in it. In our chemistry plasmas, we have lithium heparin or sodium heparin. In our hematology, we have EDTA. In our chemistry gray tube, we have sodium fluoride and potassium oxalate. Remember, sodium fluoride is the additive that prevents the breakdown of glucose or glycolysis. And the oxalate portion is the portion that prevents the blood from clotting. Maybe we need a little break. Okay, we're back. Now on to step 5, anticoagulants versus clot activators. This is also very easy. Why? Because we only have one little category that allows the blood to clot. That's our red category. Red glass, red plastic, tiger top, gold, SSTs, and the orange thrombin tubes. Those tubes will form a clot. Every single other tube we've talked about is an anticoagulant. So if it forms a clot, the end product is serum. That's the liquid portion of the blood after the blood has formed a clot. Now, if it is preventing the clot in some way, you have plasma. So every other tube is a plasma tube. In our red section, our chemistries, these are the only tubes that form a clot. So our product is serum. Now associate the word clot with serum, and then you pretty much have this down. Pretty easy, right? Let's move on to step 6. How anticoagulants actually work. We have 2 steps here. Part 1 is the ABC3 rule. ABC stands for anticoagulants that bind to or inhibit calcium. So here we have a coagulation cascade. Now what happens up here at the very top where you see factor 12 here, when we have injury to a blood vessel, some sort of a damage like a cut on our skin, this is what happens. This factor turns into this factor to this factor, and all these things have to take place in order to form a stable mesh clot of fibrin. So if one of these things does not take place, then we can't form a clot. Back to this, we have tubes that bind to or inhibit calcium. So 3 tubes actually come in here and remove the calcium or prevent it from activating, which pretty much puts a stop on the rest of this coagulation cascade, thus preventing the clot from forming. So we cannot have a clot. Now, the other part of the ABC 1 to 3 rule is that 3 anticoagulants do this. 3 anticoagulants bind to or inhibit calcium to prevent the clot. Those additives are sodium citrate, EDTA, and oxalate. All right, now for the next part, the thrombin inhibition. There is another way for the tubes to prevent the blood from clotting, and that is by inhibiting or stopping the conversion of prothrombin to thrombin. Now when we do that, we pretty much put a stopper on the coagulation cascade right here, which will not allow the blood to clot. Remember, we have to have all these things in place in order for us to form a stable mesh clot. Those tubes are our heparin tubes. Both heparin, lithium heparin, and sodium heparin prevent the blood from clotting by inhibiting the conversion of prothrombin to thrombin, thus preventing the clot. So I have a question for you. Are clotting factors found in A, clot activator tubes, or B, anticoagulant tubes? I'll give you just a second to answer this. If you chose B, anticoagulant tubes, you are right. But how does that make sense? Remember, looking back at our coagulation cascade, all these things need to happen to form a clot. Now, once this clot is formed, it can't be reversed. So you don't have any clotting factors in a clot. It's sort of like looking at a bunch of Legos. These are our clotting factors. We are aimed at building a house out of the Legos, just like our blood is aimed at building a clot when we need to. So when we form our Lego house, these are all the building blocks for our clot, but the end result is our house. So if we come down in here and let's say we inhibit the conversion of prothrombin to thrombin, we have our stable mesh clot up until we stop a part of this clot from happening. And that just sort of takes away our roof. So we don't have a house anymore. We have the building blocks for the house, but we don't actually have a house. We need all these things in place in order to make a house, and we don't take the house apart. It might fall apart because we're either stopping the blood from clotting by removing calcium, or we are stopping the blood from clotting by inhibiting the conversion of prothrombin to thrombin. So remember, our ABC 1, 2, 3 rule is sodium citrate, EDTA, and oxalate prevent the blood from clotting by removing calcium. 1, 2, 3, ABC. C starts with calcium. It's the third letter in the alphabet. All you need to memorize is which tubes do that. The second set of tubes, the ones that prevent the conversion of prothrombin to thrombin, are the green tubes, our heparin tubes. Okay, so on to step 7. There are many, many tubes, and this is why knowing the tube by their additive in the order of draw is the most important thing. Let's take these tubes, for example. These tubes have a royal blue top. There are 2 fundamentally important things that make these tubes what they are. The first thing is they belong to the toxicology department. The second thing is they are trace element free. So all of our other tubes might have trace elements such as zinc, copper, arsenic even perhaps. So these tubes can test for all of those things because they don't have any of them in it. Does that make sense? These are trace element free. That's why they go to the toxicology department. So we can test for arsenic poisoning, drug poisoning, things like that in the toxicology department. But they have to go in the order of draw somewhere, so how do we know where to put them? It doesn't make sense if we say yellow, light blue, red, green, lavender, gray, because these are dark blue, royal blue. But look at the stickers here. We have one that has a red sticker and the other one that has a lavender sticker. You guys with me so far? So what do you think this red sticker has? Well, it's a clot activator. This is going to allow the blood to clot, so we're going to get serum. This is a serum toxicology tube. What about this lavender sticker over here? That has EDTA in it. So when we look at the order of draw, now look, we have a royal blue top in our clotting activator section of our chemistries. And then we also have a lavender royal blue top in our EDTA, in our lovely lavender section. So you have to look at the label and determine what the additive inside the tube is before you can just draw it, because you cannot draw the tubes out of the order of draw. Samples will become erroneous. They will not be able to be tested. Next on our list of other tubes we're going to be talking about are the ACD tubes, otherwise known as acid citrate dextrose tubes. These belong to immunohematology and test for things such as ABO grouping and RH typing. There are 2 types of blood tubes. We have solution A and solution B. Solution A tends to be the larger of the bottles. They come in 8.5 milliliters, where solution B comes in 6 milliliter bottles, and they're a little shorter. The actual additives in these tubes are citric acid, trisodium citrate, and dextrose. As you can see, solution A comes in slightly larger concentrations, and solution B, being the smaller tube, comes in slightly smaller concentrations. We've shortened these 3 additives down to just acid, citrate, and dextrose. So keep in mind we have another tube that looks very similar to these tubes in the blood cultures category, our SPS tube. Remember, the SPS tube comes first in the order of draw, but the ACD tubes come last. So if you can remember that yucky little boys rarely get lovely girls, yellow comes first, yellow also comes last. But always double check your tube labels to make sure that you are drawing the right tubes. Step 8. Now we have another set of tubes that we sort of mentioned before, the blood cultures, but they also have their special order of draw. You'll notice on the blood cultures bottles that one says aerobic and the other says anaerobic. So what does that mean? The aerobic bottle is able to accept oxygen, where the anaerobic bottle has to be oxygen free. Which one do you draw first is all about what system of needle you are using. So if you are using the butterfly, what does this butterfly tubing have in it? Nothing but air. So the order of draw for the butterfly is aerobic and then anaerobic. Because as you can see here, the first thing when we get this needle into the arm, the first thing that's going to be coming in the tube is air. So this air is just going to be flowing into the tube. That's why we need the aerobic bottle first. Now, it's just flip-flop with the syringe. We go anaerobic to aerobic. Now, why is this? When you are drawing up blood into a syringe, you can see the very proximal part of this barrel here has a little bit of air in it. This is very common. So the anaerobic needs to go in first because that is the first thing that's going to go into a culture bottle. air will be last. That's why the aerobic tube is last when you're drawing a syringe. I really hope this helped you guys. If you have any questions, you can feel free to shoot me an email, and please like and comment on this. If you found this video helpful, you can pass it around to your friends and stuff. You can also check out our Phlebotomy Masters group on Facebook. I'll have the link below in the description. Good luck in your test-taking adventures and your phlebotomy adventures, and I look forward to hearing from you.